Okay, so I will be talking with you about the mechanism of interference by small RNA. So that was where we have ended in our last class. That the people were wondering that how actually small interfering RNA able to silence a particular gene, able to silence or knock down a particular messenger RNA. I also wanted to share some other slides as well, but I will be explaining the mechanism first, and then I will go back to the earlier slides to explain you something. This one later slide I want to show you. I mean, this is a little weird because I have added that slide last year to my medical biotechnology class, and at that time I was trying to search the examples where small interfering RNAs were utilized to deal with a particular disease. And I found this very interesting example of SARS at that time. And the SARS outbreak was there in China and in Hong Kong at that time. It created a huge fear. I mean, the coronavirus was nothing as compared to the mortality rate of the SARS. The SARS mortality rate is much, much higher than the coronavirus. But the good thing of the SARS virus at that time was that it requires an animal intermediate. It was not person to person. If you remember the President Trump's yesterday's press conference, I don't know why I used to mention that man again and again, but I mean, since he's like president of the most powerful nation in the world, so he said that he is going to block and stop the funding which United States is providing to the World Health Organization. It's very serious threat. Because the World Health Organization misinformed United States of America about the severity of the disease, and he specifically mentioned that World Health Organization said that this virus is not communicable between people. It's not person to person. So imagine that the President of United States is furious that he failed to understand this critical characteristics of that virus. So I have added that slide last year to show an example where RNA interference can actually cure a particular disease. And if you people can remember the question answer session, and that question answer session, in that session, if you can remember, I can recall my mind. Farhan asked a question, a very wonderful question, that is that possible that we can use small interfering RNA to kill the coronavirus, because coronavirus is an RNA virus. So is that possible that we can supply interfering RNAs which can go and interfere with the RNA of the coronavirus and can downplay its function? And here this is an example of that example. This is an example of that function where scientists tried to downplay the function of SARS virus using RNA interference technology at that time. And also there was a big question which was raised at that time, and which we actually facing. That question was raised like 7 to 8 years ago, but we are facing it today. That what is the killer virus that might sweep the globe? I can tell you the SARS-CoV-2, which means the coronavirus, the commonly called as coronavirus, will not going to create a doomsday. Okay, so you will survive. It will not going to create, it will not go to sweep the globe, but it actually shakes the world. It actually let us know that how powerful a one particular virus can be. I mean, imagine the countries choose to go for wars. The countries choose to make their relationship with the other countries for one single reason, and that is money. And that virus actually destroyed the economies of almost every country present on the planet Earth. Destroyed it. And it will take a long time. It will going to take a long time for all of us to recover back to normalcy after that. So that was like the thing which I have shared last year. And I was talking on these things. So let's start with the mechanism of the procedure first. That how a double stranded RNA, when entered into the cell, is actually managed to silence a particular specific gene or is able to destroy a specific messenger RNA. So once a double stranded RNA entered into a cell, it is taken up by a dicer protein. You need to always remember that thing in your mind, that the dicer and the other systems which I will be talking after that, they belong to the natural defense mechanism of our own cells. It is their natural job to seek the nucleic acids, the free nucleic acids present in the cytoplasm, because the free nucleic acids should not be there. They might belong to virus, they might belong to any bacteria, they might belong to another species. They should not survive in the cytoplasm. So their job is to seek and destroy any free nucleic acids out there. So if a double stranded DNA or double stranded RNA is there, either it belongs to a virus or a bacteria, or it is injected artificially by humans into a certain cell, that double stranded RNA will be captured by the natural defense, the machinery of the natural defense mechanism, the first member called dicer. Their job is to cleave that double stranded RNA to remove the threat. So imagine if this is a double stranded RNA belongs to a virus, the job of the dicer is to split it. So that virus will not going to utilize its gene. That virus will not be utilized to produce its protein, and that virus will not going to affect or infect that cell. So dicer will destroy that double stranded RNA into 21 to 25 nucleotide long strands, leaving 2 to 3 nucleotide overhangs. Remember, there are 2 to 3 base pair overhangs are out there. These two strands are not exactly parallel. Can you see the overhangs at the ends of each of the small interfering RNAs? Once they are like 21 to 23 base pair small interfering RNAs, they were loaded on another system which is called RISC, the RNA Induced Silencing Complex. It's a common name. Since it's a complex, it is made up of different proteins. Okay, it's a complex, so it is not one protein, it's like a combination of different proteins. The RNA, the small interfering or RNA induced silencing complex associate themselves with that small interfering RNA like this, and then it unwinds the double-stranded RNA. It actually unwinds and destroys one of them, leaving one strand kept associated within the grooves of the RNA-induced silencing complex. So one out of the 2 strands survives in the RISC complex. Just try to stay focused with that point, because if you want to destroy a particular messenger RNA, all you want all you expect that a complementary strand should survive here. A complementary strand should survive here. I will try to explain that point in more detail in the next slide. Once the double-stranded 21 base pair small interfering RNA unwind itself and one of the strand got lost in the meanwhile, you can see the red one is not there anymore. Only the blue strand survives. And the activated complex not only unwind that thing, but also helps. Where does the risk come from? Okay, Shaista, you can keep the questions, but that's fine. The risk is a part of Shaista asked a question, and I'm adding that question right now here. The risk and the dicer, they belongs to our natural defense mechanism and our machinery which controls the expression of the genes. The job of the dicer is to chop off the double-stranded RNA, and the job of the risk is to utilize these small interfering RNA for the purpose of silencing. They are natural components of our cellular systems. So after the job of the risk, we are left with one strand of that. We are left with that one strand loaded on the activated version of the risk, and that one strand which you can see is a blue one. You can ask me that why not the red one, or what happens if the red one survives. Okay, but for the time being, once again, the double-stranded RNA chopped off by the dicer, loaded on the risk, unwind itself, and then one of the strands survives along with the protein complex. The protein complex is required not only to unwind but also to keep that 21 base pair single-stranded RNA to survive, because it is highly unstable. After that, that single strand of messenger RNA, which is complementary to one of the messenger RNA out there, will bind with the messenger RNA and then destroy that messenger RNA and to silence that particular gene. Now it depends on the nature of the double-stranded RNA which you are using. If you want to destroy, if you want to silence, if you want to knock down a one particular messenger RNA, you need to design your double-stranded RNA in such a way that it should be cleaved into fragments. Those fragments then loaded on the risk, and then one of them is gone, the other one should be left with a complementary sequence of a messenger RNA of your own choice. If you made a right choice here, if you create the design perfectly, you will be able to silence one particular messenger RNA. And that is how that technique works. Once the messenger RNA found the complementary sequence, it will block the function of the messenger RNA by two means. The first, it actually destroys the messenger RNA. Once the messenger RNA do not able to execute the function of translation, this binding leads to the destruction of the messenger RNA, because messenger RNA is supposed to be very delicate. The other way is that as long as the risk proteins are out there on the messenger RNA, it will not allow the ribosomes to utilize that messenger RNA anymore. So it can actually block the translational machinery as well. So as long as it is there, the translational machinery will not be able to translate that messenger RNA, and that is how it actually blocks the function of a particular gene. In nature, that mechanism is also used to control the expression of certain genes. And what we have discovered, since we have discovered that mechanism, that how nature controls the expression of a certain gene, we started mimicking that process. Whenever nature wants to control the function of a certain gene, the nature wants to downregulate the function of a certain gene, Nature creates a double-stranded DNA or double-stranded RNA in the system, which is chopped off by the dicer and produce a small interfering RNA in the same way. So it is either provided by you or it is either created by the nature. To do what? To control the expression of a certain gene. That dicer created the small interfering RNA, which will be then loaded on the RISC. Actually, there are certain other proteins. The Argonaute protein is there as well, which I will going to show you in that video, and I will explain that video myself. So that RISC, like loaded with a small interfering RNA, unwinds and then left with one strand of that small interfering RNA, which is complementary to the target messenger RNA. Once that complementation is there, that messenger RNA is doomed. It will not be able to execute its function. The gene is silenced. So this is the way the nature can downplay the function of a gene, keeping the integrity of the genome. So there will be no effect on the genome at all, but the genome will not be able to execute a function. And this gives the diversity in functions between different types of the cells. And another molecule which is created naturally within the cells, they are called microRNA. MicroRNA are also the small RNA. Usually they are single-stranded RNA, small RNA, but if they have a self-complementation options, if they can complement each other, a single-stranded RNA can create this structure with a hairpin shape. It's called hairpin loop structure. If they have certain base pairs complementary with each other, this is a single RNA, but some of the base pair matches with each other, and that is why it creates that structure. This hairpin structure helps that small RNA to survive for a little longer period of time, but it can also captured by the dicer gene to produce small interfering RNA, which can later finds the target messenger RNA to down-regulate the function of that gene as well. This is another natural way of controlling the expression of gene. The naturally occurring RNAi molecules, the microRNA, they are like the master regulator of the expression of genes. So it came from the genome. Of course, it came from the genome. They are produced by the RNA polymerase, but their RNA polymerase is different. The messenger RNA is transcribed with the help of RNA pol 2, but the microRNA are transcribed with the help of another RNA polymerase called RNA pol 3. So they came from the regions of the genome which are not producing any protein. So they are also genes, but they are non-protein producing genes, or you can call them as a non-coding genes. So once they are there, like here you can see in the system, they were chopped off and captured by the dicer, and then the RISC complex with the Argonaute proteins, and then one of that strand is utilized to downplay the function of a certain messenger RNA. The difficult thing which you should remember for this procedure is that if even at that place, let me show you in the earlier slide, even if few base pairs of a small interfering RNA came from the microRNA, this small interfering RNA came from the microRNA, even it managed to find few base pair complementarity, like 5 to 6 to 7 base pair complementarity with the microRNA. The microRNA produces the small interfering RNA, and that small interfering RNA finds few base pair complementarity with the target messenger RNA. if it happens, it can block the function of that gene. If it happens, it can block the function of that gene. Now imagine if that one microRNA at the same time able to find complementarity with 5 to 10 genes, with multiple genes, like 6 to 7 base pair complementarity with one gene, 5 to 6 base pair complementarity with another gene, 5 to 6 base pair complementarity with another gene. So in that case, one particular messenger RNA will be able to control the function of many genes. And that makes messenger RNA very unique and very special. That even one messenger RNA can influence the expression of many genes and can change the function of and can change the transcriptome of the cells to a large extent. You can see here the dicer proteins are present naturally within the cell. They will work whether the double-stranded RNA is provided by you, or is provided by the virus, or is provided by the microRNA, or whether it is provided by the non-coding RNA. The dicer proteins don't know from where these small double-stranded DNA or RNA are coming. They can come from different sources. And the dicer will going to chop them off, send them to the organoid protein system or the RISC proteins, and then they will execute their functions. They might degrade the messenger RNA to block the function, depending on the nature of the things which are coming from upstairs. They might inhibit the transcription. They might change the transcriptome and the cell fate. So nowadays, a lot of people are working, a lot of research is going on that point, that the cell fate or the nature of the cell, it depends on the patterns of the expression of the genes. Who is actually controlling the pattern of expression of the genes within the cell? So one particular, you can say, culprit, you can pinpoint, might be the microRNA. And that is why the microRNA is a very actively researched species nowadays. The people who are studying cancer, who are trying to find the reason behind the abnormalities of normal cells, they are looking that it might because of the change in the expression of the microRNA within the system. It also gives an opportunity for all of us and for the researchers to use microRNA for a therapeutic purposes as well. To use the small interfering RNA to dilute the function of a certain gene, to silence or knock down the function of a certain gene. And that is happening as well. This is what I wanted to show you earlier, that where we are using the RNA interfering technology, we are using it to study the function of a gene, to identify the pathways, to study the role of microRNA for the expression of certain genes, and we are trying to cure cancer and trying to control the viral infection and parasitic infection with the help of RNA interfering technology. Okay, so this is it for the first session. I will going to conclude that talk in the next section, and we're going to take your questions as well. Here we go. So in order to conclude the session, I would like to take the help of a video which I have already shared with you, and I believe that you people already saw that video, but just have a look in that video. I will be asking some questions from you as well. And I think there was a very funny little question about it. And I have shared my screen with you as well. And let me start with that video. It's a very beautiful video, wonderful animation created by the nature. So I don't think you will be able to listen the voice, so you only depend on the voice from your instructor. This is the universe of the cell. This is like the planet cell, I must say. It has its own world, it has its own creatures, and it has its own rules and principle. So we are going to zoom the cells. We are going to enter into the planet of the cells from outside. So here we go, the journey begins. So we are entering into the cells. We can see different components floating here and there. And these components which are floating, they are like the proteins present in the cytoplasm, different enzymes executing their functions. And here we are right inside, moving through the cytoskeleton of the cells. And my question, first question with you is that I will see which one of you will be able to tell me the name of this lazy stuff. Can you see the Humpty Dumpty? The Humpty Dumpty sat on a wall, something like that. Look at the movement. Iski chaal dekhiye aap. You need to tell me the name of that thing, okay? This beautiful goal-matol stuff which is walking on something, and you will let me know what is that something and what is that protein which is moving on it, okay? Later on you can answer me. So we are looking at the beautiful landscape of the cells. We are moving through cytoplasm, and here we go. We started looking at the nucleus. Oh my God. And you can guess what are these things? These holes, these are the nucleopods. From where whatever the material is created. So whatever the material is created. तैयबा आप इसको तैयबा ने आंसर किया है कि ये आरएनए है उनके ख्याल में. आप इसको संभाल के रखें अपने आंसर को, ये ठीक भी हो सकता है. It might be a bingo. Okay, I will be asking after that. Let's stay in the story. Okay, so look at the double membranous bounded core of the cell. This is the nucleopode. Okay, I will be taking your answers. Okay, Shaista, hold on, keep your answers. Okay, after that video, right after that video. So we will be looking at the nucleopodes, whatever is created within the nucleus, especially the RNA, whether they are small RNA, they are long RNA, messenger RNA, micro RNA, they are required to be exported into the cytoplasm. So they are Galaxy C7 Pro. I wish that one should be our student. So these nucleopods are required to export the things into the cytoplasm. So what that means? That the microRNA or the non-coding RNA will be traveling out of these nucleopods into the cytoplasm. Even the messenger RNA, it is translated in the cytoplasm. Okay, so let's move a little further. Nucleopods, look at that. What is that thing? This is the genome. This is our DNA. And that DNA is used, the strands of DNA is used to create what? To create the RNA with the help of an enzyme called RNA polymerase. If this is a messenger RNA, synthesis is going on, then this one should be the RNA pol 2. This is the strand of DNA. This is the RNA polymerase. And here we go. This one, this thing which is coming out. This is the newly created RNA coming out of that thing. All these little small components which are entering into the RNA pol 2 are the nucleotide bases, which will be incorporated to create this thing. So once the messenger RNA is created with the help of certain supportive proteins, you know what's you just saw something very amazing. Let me repeat it for you. This thing and this thing and this thing. See it again. This is called RNA splicing. The pre-messenger RNA is then spliced, and then exons were removed and put together to make the messenger RNA. And this machinery is converting the pre-messenger RNA into a functional messenger RNA. Look at that once again. These people have done a wonderful job putting these fragments together and removing the other one, okay? And after that, you know, it will be exported through the nucleopores into the cytoplasm. So now that messenger RNA is into the cytoplasm. In the cytoplasm, that messenger RNA is utilized by the translational machinery. This is the ribosomes, 2 fragments. You can see the smaller one here, the bigger one here. Amino acids are supplied by whom? The amino acids are supplied by the transfer RNA. This is the transfer RNA. And you can see this thing, the anticodon, the famous things you already know. So these transfer RNA brings, they brought the amino acids within the ribosomal machinery to create the proteins, and that is how the proteins are created. So every messenger RNA is translated within the cytoplasm if it contains all the necessary information. And you know what are those informations? The messenger RNA should have a poly A tail. The messenger RNA should have a 5' cap. It should have a 5' untranslated region and a 3' untranslated region. It should have a start codon. It should have a stop codon. All these important little information should be there on the messenger RNA. So the translation machinery should be able to recognize it and should be able to produce protein out of it. But what if we are supplying with a double standard DNA or a double standard RNA? So this is injected into the cell by humans or by a virus. What if we provide that double standard RNA into the system? When that double standard RNA present in the cytoplasm, it will be sensed by the cellular defense mechanism. Or the second choice, the second thing is that if a microRNA is created from the nucleus, the nucleus means the DNA create the microRNA for one purpose, and that purpose is to control the expression of the gene. You have provided that double standard RNA to do what? To control the expression of the gene of your own choice. So what we are doing, that we are trying to steal the capability of cells' natural mechanism. What is the cell's natural mechanism to control the expression of gene? Is to produce messenger RNA, is to produce microRNA from the nucleus and then export those microRNA into the cytoplasm. So these are the microRNA supported by the supported proteins, and they are entering into cytoplasm to do what? To control the expression of a certain gene. So either the microRNA. This one is the microRNA coming out into the cytoplasm to control the function of certain genes. Or it is provided by us or humans, like a double-stranded RNA is provided. Both will be treated in the same way. This is the natural way of controlling the gene expression. And this is the human way of controlling the gene expression by giving them the double-stranded RNA. The double-stranded RNA which you have given and the microRNA will be processed in the same way. You will see how. First, the disoprotein will capture and it destroys the double-stranded RNA into small fragments. And they destroy the microRNA as well into small fragments. And you know the length of the fragments, about 21 base pair to 23 base pairs, with 2 to 3 base pairs of overhangs. Then loaded onto the RISC Argonaute protein. The Argonaute protein not only unwinds the double-stranded RNA, but also leaves one of the strand out there, which is destroyed by the other RNAs, enzymes present within cytoplasm. Now the Argonaute protein is left with just one strand, and that one strand is complementary to one of the target messenger RNA. This is the messenger RNA, and the Argonaute protein with that one single strand which is left will be able to bind with the target messenger RNA. This one strand of the small interfering RNA present in the Argonaute protein is called as the guide RNA. This is a very critical information for a new technology which develops after that. This one RNA strand is called as the guide RNA. The Argonaute protein or the RISC complex don't have eyes. They don't know where to go. They don't know their target. The target address is coded into that small interfering RNA. It will take the Argonaute protein to the destination. And this is the same technology, similar technology will give birth to the CRISPR-Cas9 system. And we will study that system as well. It has the same type of story, that an enzyme knows how to cleave the DNA, but that enzyme do not know where to go. That enzyme is blinded, unguided, and you gave something to guide that enzyme to reach at the place of your choice. So that small interfering RNA will take that Argonaute protein to the target messenger RNA, and after that, that binding results in the degradation of that messenger RNA. So whatever the nature of that small interfering RNA is there, it will going to influence the capacity of messenger RNA. It will go and destroy the function of that messenger RNA, and that's what we call as gene knockdown. And that occurs after the transcription. That occurs once the messenger RNA is created. It is always reversible, because if the small interfering RNA, you are not providing any more small interfering RNA, you will not be able to silence more genes after that. So that is how, as long as the small interfering RNAs are there, the messenger RNA function will be compromised. The cell will not be able to produce that protein, and ultimately the cell will lose the function of that gene, and this is what we call as the gene knockdown. The beauty of that procedure is depend on the potential of human, the potential that we can design the double-stranded DNA with our own choice. That means we can choose which gene we want to silence. Okay, so and also before I conclude, I also want you to look at the stories in a more scientific way, that people do not end with the final story in one go. It always proceeded gradually in the form of stairs. Like we discover one thing, that will take us to the next, and that will take us to the next. So it's not like the RNA interference machinery started affecting science and research immediately. People tried that technique to interfere the human immunodeficiency virus, the HIV virus first. And that was the exact question the Farhan asked on the other day. Can we interfere with the coronavirus? The answer is yes, we can interfere. But the nature of infection is very high. And the latest video which I have shared with you on the YouTube channel, the paper which I have explained in Urdu, that shows that how coronavirus is able not only to infect the lung cells, but it is also affecting the T cells as well. Like the HIV virus. The HIV virus do the same thing. आपने सुना होगा कि HIV virus क्या करता है? वो इंसान का immune system compromise कर देता है. Somehow scientists has discovered that the coronavirus is also affecting the immune system, as that is why it is very contagious and very much infectious. And it was published in the last week in Nature. So if we are like studying that information in the textbook, we will read the same information after 7 or 8 years, after 5 years. तो पाकिस्तान से इल्म गुजर चुका होगा, दुनिया उसको पढ़ के जा चुकी होगी जब उससे पढ़ रहे हो. If we try to understand that thing today, that means we are living with the rest of the world. And remember, the next universities will expect you to understand whatever the rest of the community, scientific community is doing at this time. Now, you need to be updated as a researcher or as a scientist. Like the stories evolve in this way. I have placed these different papers together. So you will be able to see that how RNA interference technology is utilized to work against a certain virus or to work against a certain cancer. So all these things were working in parallel at the same time. Okay, so now I'm done. So the floor is available for your questions. So you people can ask your questions, and I'm still sharing my screen, so I might be needing some of the slide with you. Okay, so it's wonderful if you can speak. It's all right. I mean, अगर आप बोलना चाहते हैं तो आपको सिर्फ अपना mic unmute करना है, and you can speak as well. But if you want to write your questions, you are always welcome for that thing as well. Okay. So तैयबा ने आंसर किया इसके बारे में कि RNA है, जो मैंने क्वेश्चन किया था lazy daisy, and then शाइस्ता ने आंसर किया it might be a ribosome. No, शाइस्ता ने आंसर किया a ribosome. Zainab ne bhi, I'm not answering you, okay? I'm giving you the time to find your answers, okay? I'm just taking your answers right now. So Hadia ने अपना problem बता दिया मुझे कि sir I have a class at 12 of molecular biology. Of course, I understand. I know I should find another way for that thing. क्योंकि हमारी 11 बजे मीटिंग होती है यूनिवर्सिटी फैकल्टी की. I don't want to speak on these things. I mean, it's a recorded lecture. I will try to fix that thing for you. Okay, I have to edit that clip of that class issue. I will try to fix that thing. Okay, Ramsha, I note down your point. Then Raha asked G-actin subunit, and then Farhan, that was the kinesin protein walking along the microtubule or the microtubule in the video. Okay. Hamza ne bola ke motor transport proteins hain. Okay. So these are the answers of that lazy daisy. Zainab, you want to speak you want to ask something? Please go ahead.
Yes. So you mentioned that the siRNA and mRNA, they're processed in the same way. So like, is there any competition between the two when if they're complementary to a singular sequence of an is the same?
Sorry, Zainab, I failed to get your question because of internet speed. I think it's from my side or maybe from your side. You need to repeat your question, okay? Please repeat your question.
You mentioned how the siRNA and the mRNA, they're processed in the same way by the dicer protein. So will there be any competition between these two types of RNA if they are complementary to the same sequence of the target mRNA?
Of course there will be. Of course there will be, because the dicer proteins don't care what will happen after that. They will just chop off whatever they are getting. The risk proteins don't care what will happen after that. They will try to unwind whatever the things they are getting, and they will try to take it to the target places. So if the target places like are lot, जो कि बहुत सारा होती हैं, because the messenger RNA copies are like huge in number, so they will be able to destroy the messenger RNA separately. But if, of course, if there is like 2 messenger RNA out there, and there are 4, double-stranded RNA and 4 microRNA out there, so there will be a competition, of course. The answer is yes. G.
Thank you, sir.
हमारे पास और if you can speak as well if you want to ask a question. I have, okay. I wanted to ask how the cell reverts the RNAi mechanism. Are there other regulatory proteins involved in this? Can they, sir? Okay. So it's a good question, Raiha. कि cell इससे जान कैसे छुड़ाता है? If the RNA interferences molecules are there, it will go and silence the function of a particular gene. The answer is yes. It's like a disposable stuff. So if we have like 10 interference molecules out there, they will go and destroy the function of the messenger RNA, and then they are consumed. Finish. The new messenger RNA will not going to be compromised because there is no small interfering RNA left. There exists no extra small interfering RNA out there. So as long as if we want a certain gene not to express, we have to provide a consistent supply of small interfering RNA in the system. So as long as we are providing the small interfering RNA, that messenger RNA will not be expressed. But once we stop the supply of small interfering RNA to the cells, the function of that gene will be restored because nothing is there to stop that function. I want you to see a little advanced version of whatever I am speaking. This slide, you need to see that slide. In this slide, people have incorporated a certain sequence into the genome of human cells or mammalian cells. So this is a gene construct like a vector. This is the vector. It contains important components like it has the EGFP reporter, so we can see whether it is incorporated or not. It has a promoter, human promoter as well, mammalian promoter, U6 promoter to produce small interfering RNA or to produce small microRNA. And then the SIN LTR. These sequences are used to integrate an external DNA into the mammalian genome, into the human genome. So if you create that vector and you add a certain gene, for example, not a gene, if you add a certain sequence of a nucleotide incorporated into that vector, that part of the DNA will going to be part of the human genome as well. Now this U6 promoter will continuously produce that double-stranded RNA, stem loop sequence, will continuously produce that 21-23 base pair of the stem loop structure. It will continuously captured by the dicer. It will continuously kept on silencing the target gene forever. So as long the human genome contains that vector, that portion, that gene will not be, the target gene will not be expressed. So this is like a stable way of silencing a gene within the cell forever. By supplying the source, by supplying a vector which can produce that stem loop structure as long as the cells survive. The other way, the temporary way, is to keep giving the small interfering RNA to the cellular system. So as long as you are giving, you are sending the small interfering RNAs, you are down regulating the function of a gene. Okay, so this is a very advanced method that if you want to a certain gene not to express at all, you have to put that vector which will keep providing the microRNA or the small interfering RNA so that it can permanently block the function of that messenger. So as long as the cell lives, its DNA lives. If its DNA lives, that vector survives. That vector survives, it will kept on producing the microRNA, and that microRNA will kept on suppressing the expression of the target genes. Okay, let me see other question. So Shaista asked a question that can dicer chop off microRNA and small interfering RNA? And the answer is yes, Shaista. Both of them can. One dicer will not be able to chop these two together, but the two different dicer proteins can chop off the two different components at the same time. Taiba asked another question. Despite the knockdown of gene, cell can also use it as a control for regulation of its protein product. Despite the knockdown of a gene, cell can also use it as a control for regulation of its protein product. Taiba, let me try to understand that question. Although the gene is not there, it can knock down the gene. Cell can also use it as a control for regulation of its protein product. Of course, cells use the microRNA to control the expression of the genes. So that is how the microRNA and small interfering RNA works. Small interfering RNA or double-stranded RNA we are providing to the cells to downregulate the expression of a gene. MicroRNA are produced by the cells, so they can control the function of a gene. So both of them are yes. It's more like a comment. If you want to ask more, just please write in a little bit more detail. And then Shiza asked a question, which factors decides that out of double-stranded RNA, which strand will survive? Oh my God, this is a very important question. This is actually a very technical question, and I will be asking that question in an assignment. Okay, because when you design a double-stranded RNA, what you should have in your mind, you should have that thing into your mind that at the end of the day, at the end of the story, the blue one, the strand of RNA which left at the very end should be complementary to the messenger RNA. If it has the same sequence, then the messenger RNA, it will not bind. Think about the two strands, the blue and the red. In this case, the blue is the complementary sequence. That means the red one is the same sequence as the messenger RNA. Now, in order to design that double-stranded RNA, you need to know certain points. That you need to know which side or which strand will going to be cleaved in which way. There, I can give you a hint before that assignment. I am giving you a hint that I have shared certain components like this on a vector. If you can see here, certain components. CMV is a promoter which can express a mammalian gene, and here it will express EGFP. SIN, SIN, they are DNA elements. LTR, long terminal repeats. Where these type of DNA is present, it will help the vector to incorporate into the genome. So every DNA element have a certain function. U6, a promoter. So whatever after that will be assumed as microRNA because it is RNA pole 3 promoter. CMV is a RNA pole 2 promoter. So whatever is after that should be a messenger RNA, should produce a messenger RNA. So a similar type of signal should be present here, which should define which will be the +1 side and which will be the tail side, and which of the side will be left at the end of the day. Okay, so you might need to search and find that answer for me, but it's a very Good question. And I saved that question to create an assignment for you. Okay. So Shiza Shabash, very good. Farhan asked a question. Could guide RNA be targeted for degradation by the RISC complex as well? We don't need to destroy the guide RNA. Actually, it is the job of the guide RNA to find the complementary sequence on the messenger RNA. So destroying the guide RNA is not make any sense, since it's a very small fragment of RNA and it is created out of the double-stranded RNA. So we are not targeting, we do not target the guide RNA because it's a very small fragment of RNA. Okay. Anybody else wants to ask question? Hamza, जी Hamza.
सर आपने मेंशन किया था कि रिस्क से पहले ये एक और प्रोटीन के साथ एसोसिएट हो जाता है जो हमारा सिंगल स्ट्रैंड होता है आउट ऑफ द mRNA या siRNA. So when we introduce this kind of double-stranded RNA into the cell, तो हमें इस तरह की प्रोटीन के लिए वो स्पेसिफिक गाइड RNA भी प्रोवाइड करना जरूरी नहीं?
No, हम लोग जब डिजाइन करते हैं डबल स्ट्रैंडेड RNA को, जिसको हम इंजेक्ट करना चाहते हैं, when we want to design small interfering RNA, we only consider one important component, जो शिजा ने पूछा. We just need to make sure that whatever the cellular machinery will do, at the end of the day, the RISC should carry that strand of RNA complementary to the target messenger RNA. That is the, like everything we want to. The cellular machinery is blind. The dicer is not able to distinguish between the source of the nucleic acid. It is just designed to chop off any nucleotides out there. So whatever you are creating will be destroyed for sure. But you will create the things in such a way, it's your job. That is science, that is research is all about. You will design double-stranded RNA in such a way that it will give a product which can silence the gene effectively. And I can tell you that many people, most of the researchers who are working in the gene knockdown, they are making this mistake, that they are actually trying to create a small interfering RNA, and accidentally they ended up with a DNA, they ended up with a single strand with the same sequence of the target messenger RNA, and they failed to find any knockdown. Nowadays, since we have evolved that technique a lot, so it is not your problem anymore. So I'm those type of instructors who wants you not to consume your brains for the things which you can do like with the help of technology. Like you are not supposed to multiply with the traditional method. If you have a calculator out there, you should save your time. So now we have portals out there, and this is also part of the assignment which I will be giving you with this topic. Those portals ask you that what is your target messenger RNA, which gene you want to silence. All you need to do is to give them the sequence of your gene, and based on the components and research and the signals, they will give you the sequence of your double-stranded RNA or the double-stranded DNA. They will give you based on all the laws. Okay, sir, this is something which you need to create, and if you manage to create that thing, you will effectively able to silence that particular gene. So when you order the companies the small interfering RNA, they will ask you what gene you want to silence. You just need to give them the sequence of the gene, and they will use their system to create like multiple small interfering RNA based on their calculations and algorithm that this will be more effective, like this will be more effective. They will create 4 or 5 targets, optional small interfering RNA for you. So you can pick, or you can order all of them to try. So that is how we are able to silence a one particular target messenger RNA. G. Next. Who is the next? I got the question. Let me see the question. So how the overhangs at the end of the interfering RNA helps its function? Is there any significance? Yeah, Shabash Mustafa. There is a significance of overhangs, and it's not only the overhangs. The question is quite general, and it is applicable to many other things as well. That the dimensions, the architecture of a substrate is very critical for the function of the enzymes. You know that one enzyme is only able to catalyze, enzymology mein padhte hain aap, is only able to catalyze one particular substrate only, and that depends on the orientation and the geometry and the dimensions and direction and whatever the 3-dimensional structure of that substrate, because it fits in that pocket of that enzyme. The overhangs are critical in this way for these reasons.
Sir, I joined right now. Could you repeat the answer, the last point?
Okay, my answer was that every dimension of the substrate matters, okay, so that it can effectively fit in the pocket of an enzyme of a protein. So having said that thing, the overhangs of the small interfering RNA allows that small interfering RNA to fit perfectly in the pocket of the proteins out there. So these things matter like any other substrate.
Okay.
Ji, shabash. Question nahi hai. So I would like to thank one by one all of you once again. Anisha ko shukriya bolunga. Thank you very much. Anusha is here. Thank you very much. Farhan, Hamza, Imama, Laiba, and Like is here. Thank you very much. Marwa is here. Mehreen is here. Thank you. Mustafa, thank you very much. Kadesh, thank you very much. Roha is here. Rimsha is here. Shaista, thank you very much. Shiza, thank you very much. Taiba is here. Tuba is here. And Zainab is here. I believe that there might be more people जो यहाँ पर थे और अब नहीं है. उन सब का भी बहुत-बहुत शुक्रिया. If you don't have any questions, then I wish you a very good day, and please stay safe and stay alive. Okay? I wish you all the very best and have a very nice day. Okay? Bye bye. It's bye bye from my side. See you soon.